Da.
Glad to be with you. Thank you very much for your organizing committee, which makes the honor to me to invite me. Your lovely city. It was a very big occasion for me after about 10 years that I come here to meet my colleagues. It's very important because we have the same mentality, the same scientific background. It's very important. Like before starting to thank my clinician colleagues all these years who trusted and trust me and my job and my colleagues all these years. Thank you very much for everything. So, let's speak about our conference. I'm invited from you to speak to you, to discuss with you about a very crucial field, which is the quality control of the IVF laboratory, which is our house of every day. This is our house for everybody of us, the embryologists. Sorry. There are different definitions about the quality and how the quality started to be introduced in our field, and we have to mention that the quality started first of all not in the scientific fields but in the market. So speaking about the total quality, there are different definitions, and this is a very organized definition. It is a well organized procedure which contributes to the satisfaction of the internal and external clients by ameliorating the total of the partial steps via well-programmed, organized, directed, and controlled acts. Speaking about the definitions of the quality, there are different approaches. There is one approach which speaks about the quality. Which is the quality? It's a fitness for the use. It is the adaptation to the requirements of the clients. And finally, it has to be in compliance always with the standards of the market. Concerning the management of the quality, this is a total of activities which contain the quality control, the assurance of the quality, and surely the amelioration of the quality. Looking back to the past, we have to mention that the quality management of all the products, first in the market and in the commerce, began after the Second World War with the Japanese products. And during the 60s, we have at this moment the total quality control, because this control showed the high quality of the Japanese products. During the 80s, we adopted this total quality control, and during the 90s, we have the transformation of the total quality control to the total quality management. It is exactly, my colleagues, that the total quality management, and which is what it is the scientific way of doing business. And so, from one part, the total quality management, it is ideal even for the IVF units. Quality means, anyway, in any case, duty for care and best practice. This is sure. Edward Deming said that the good quality does not necessarily mean high quality. It means anyway a predictable degree of uniformity and dependability with a quality suited to the market. This is for the commerce. But our scientific godfather, Sir Bob Edwards, pre-Nobel, during an IVF Congress in Sydney, said that time that the IVF laboratory must be run by the scientist, not the clinician. We speak about the laboratory, emphasizing anyway the importance of the team approach and the mutual respect. And he added that scientists should be more able to understand business and finance as well except the scientific field. Let's speak about the evolution of the IVF. It started in 1978, as everybody knows, Louis Brown, as a revolutionary experimental protocol. During the 80s, we had an enormous development of the research and the clinical practice. In the mid of 80s, the IVF units, clinics, are slowly characterized like big commercialized industries. And after that, till today, every day we live that in IVF clinics, we live a routine, but in a good way, with a good meaning, a routine of clinical and laboratory services, because every day have different surprises and different expectations. So the consequence is that the management of the quality control and the avoidance of bad events, which is very important for us, for the unit, for the clients, must be the main purposes of an IVF clinicians, and scientists as well. Quality control, as I told you, means best practice. What does it mean? It means efficient and safe IVF therapy, high levels of scientific and medical services, and finally what? Proof of care for patients, but for the members of the IVF unit as well. Slowly, we had the apparition in our field, in any field, of the ISO, the International Organization for Standardization. The ISO works with three fundamental principles. The consensus, which means to take in mind of all the participants' arguments. The industry-wide, which means that we have to give satisfactory solutions for industries and clients all over the world. And finally, the voluntary, which means that the market puts the standards based on the voluntary contribution of all the participants. This is the different editions of ISO from 1987 till 2000, where the quality management system is introduced. And to be more specific, we have the different adaptations of the ISO concerning the quality management systems, the guidelines for the quality and the environmental management systems, and then for the function of the medical laboratories. Speaking about the main quality control systems in our field, in the IVF units, we have to know that for the IVF unit there is a special ISO. For the embryological laboratory there is another separate special ISO, but except all this, we have to take in mind, dear colleagues, the European tissue and cell directives for the normal function of the laboratories and the units, and for each country, the national committee's technical specifications, which are needed for the license and the function of the IVF unit. In any case, the IVF units have to show a quality which corresponds to the necessities and the expectations of each patient, but at the same time, it has to offer efficient, sufficient, and safe services which protect the rights and the personality of each participant, including the newborn. So it is important to say that the scientists, us, the scientific members of the IVF unit, have to think and act as scientists, and that the management of the quality control is a part of the scientific background of each member of our unit. It doesn't belong to one person, as we understand and as we perceive. Quality control for an IVF laboratory means, first of all, high cost. It's normal. High cost, why? Because we won't require we won't require the certified equipment and highly qualified culture media. But okay, high cost, the high cost is one part, but the consequences are that the advantages, the three best advantages, best percentage of pregnancy, professional satisfaction, very important for everybody, and high financial benefits, very important for everybody as well. Benefits for the clinic, so the purpose of the quality control, we can see here, to develop and ensure the quality of the clinic, to have an harmonic function in the clinic among the members of the clinic, to avoid the errors and the mistakes, and if this occurs, to be able to identify the reasons and take decisions, take action, and finally to determine the ways to improve our activities. But we have to mention, dear colleagues, that the use of the tools, and this is very important for me, the use of the tools of management could depend on the number of persons who work in an IVF laboratory, unit, the possibility of understanding the necessity of the existence of these tools, and finally, the possibility of using these kind of tools. There are some questions which should have an answer in our document control system, such as, does the document have a unique identification every day, an issue number and a current revision status, a date of the latest issue, a pagination in order to have a good code, a good series, a good control of our results. And where can I find this document? You ask your colleague, I ask my colleague asks me, where can I find the document of this result? Okay, physical location, in which level, in the ordinateur, in the computer. Who assures that only the latest issue of the document is present in our laboratory, in this system, and who removes the outdated issues and files them? All these amendments, are these amendments to documents clearly marked in order to have the main control? And how are the changes in the document implemented with the personnel? This is very important questions, all these, for the normal control. What anyway needs monitoring? Okay, the ISO quality exists and it has there are some standards which can affect the process, which process can be monitored. First, the temperature. We have to take in mind the temperature of the room, of fridges, of incubator, of heated stages. Always control them. The pH, very, very important, as you know very, very well, in different irrelevant mechanical media. And finally, which is the final result, which is the apparition of our work. You know that doctors' embryo transfer results, not only, but in collaboration with them, for us, embryologists, IVF or ICSI results. Very important. This is there is no, the unit, you know very, very well now, it is a collaboration. There are not separate duties. All the duties are mixed. Okay. What about the tools for the control of the quality and the risks in an IVF laboratory? Inspection. What about the environmental conditions? Very, very important. You have very, very good rules concerning the air quality, the square meters, very important. In accordance with the European directives, very, very important, because for our job, for the result of our job, and for the pregnancy. This is very important for the couples. Because of that they come to us. Yes. The correct function of the laboratory equipment all the time. Be stressed about that. But if they work okay, we are not so stressed. We are satisfied. Consequence from transfers of people and materials in the laboratory. We don't want that. We don't want that. Nobody can enter in the laboratory like this. It's our decision. The suitability of the products, the performance of duties, the collection of information, the data analysis. Another tool, the failure modes and defect analysis. What's that? Okay, we have a problem, it's normal. You are human, we think. Determination of the problem is not bad. We have some bad moments. Okay, we are not like furniture. Okay, determination of the problem. We make the identification and we analyze it. We follow up and we coordinate. And finally, we have to correct, we have to find a solution, and we estimate this solution. Root cause analysis as well. Let's see. Localization of the real causes of bad results. Not like this, because I think that, okay, because it is Tuesday and Tuesday the wind is like this. No, there's a special reason for the bad results. Avoidance of the appearance of similar problems. Very important. It's not bad to have some bad events, but we don't want the reappearance. Enormous application in the field of health. You know very, very well that these fields of medicine and biology are surely very, very complicated, first. And second, that be sure that my colleagues, that the source of mistakes, the source is often not the human mistakes, but the bad functions of the systems that we use. It's very, very important. Let's see here from one study in Adelaide. Okay, it's not so recent, but it is representative. Can you see here? You can see here, sorry, the pregnancy rates before and after the ISO. You see the mean pregnancy rate here between this period, it is almost 28%, and it went to 40-45. You have an augmentation around 67%, very, very important after the ISO. But as well, what about the effects of the quality management on the fluctuation of pregnancy rates? It's sure that the fluctuations, the pregnancy, it's not a line. There are always fluctuations. They are always explications, even from the physiology, from the function of the gametes. The physiology speaks about that. We know that. But, okay, this was the pregnancy rate before the quality management. You see, okay, except this event which can exist. After the quality management, it is the green line. Always fluctuation, but in a shorter space. And when we speak about the average pregnancy rate, before the quality is the red one, and with the blue is already better. Very important. Which is the heart of our lab? Or at least one of the heart. How many hearts we have? A lot. But this is one very important, the incubator. Should be of highest quality, so it is expensive. Okay, it has to have a closed humidity system, a sterilizing program, and multiple doors as well. Look here, you know that, but just to mention, which is you see you have a 50 micron drop under oil, and we have to check the pH. Anyway, when we prepare our dishes for the next day, it takes a time time of 35, 40 minutes to equilibrate. And from the time that we put outside of the incubator for 3 minutes, not more, the dish, the pH makes a fluctuation and it needs another 10 minutes to re-equilibrate. So you can imagine if you open, if you close, the same, it is a disaster for the embryos, especially for the culture of blastocysts. So we have to have adequate equipment concerning the pH meter, always with an accuracy of 0.05, not stable. For concerning the temperature compensation, because we have to know that the pH is not the same between 27 and 37, 25, sorry, and 37 degrees. And anyway, we have to calibrate, and 3-point calibration very often in pH ideal 4, 7, and 10. Concerning the thermometers, an accuracy of 0.2 degrees and calibrated yearly, once per year, by external laboratories. What about the SOP? You know that everything that we do in the lab, it is an SOP, standard operating procedure. What's that? Somebody told us, somebody taught us, somebody explained us that we have to say what we do for the SOP. We do what we say. We have to show what we have done. And we have to make the question to ourselves. Did it end up as we thought it would? And finally, do we have an improvement? And if we have, we say again what we do. And we start again. The SOP has to define a range. As I told you, the temperature has an accuracy of 0.2 degrees, the pH meter as well, and very important before, there should be a minimum pregnancy rate for each operator. You cannot have 50% you and your colleague 10%. Something in the same conditions. Something happens. We have to work with the same unit, with the same standards, with the same ideology. For me, you know, the ICSI is the biggest revolution, the biggest invention in the field of human reproduction. We know that. And there is no other, for me, so significant invention. We have to be okay with our colleagues and our colleagues with us, that we take every step on the same way, in the same laboratory. We cannot do different things. So we denudate, we make the sperm on the same way, we observe on the same way, we write on the same way, and in order to have, at last, similar results. Personal advantages. Surely, you live them, and I live them every day. Low personal and collective level of stress. Is that true? Is that true? I don't know. After 25 years, I have the same stress every day. Warm internal flame due to the fact that, okay, you became a better scientist. What else? Be sure that you will not leave the bad consequences of the suspension of the function of the lab. Very bad. Feel comfortable to answer the question. Very, very important for the assurance. Which is the cause of a bad result? We have to answer. And finally, sleep well during the night. Anyway, what matters most is how to see everybody, how he sees himself. Okay? I have other things as well. Let's see. So the prognosis is better than the therapy. Our clinicians here have to show it and to know it. Listen, the quality management, dear friends, is an ongoing process. Even if I go from Bucharest to Athens, or I come here, but okay, handsome man, okay, beautiful woman, but look, ongoing process. Improving quality is key to future success. Yes, let's see why. We started with okay, revolutionary car at this time, but not the same with the Formula 1. The very sympathetic ladies from the church, from the monasteries, they gave us the urine. We made the gonadotrophins in the beginning from the urine. But the evolution is what? Recombined. Biology, molecular biology. We trust them for different reasons. quality management provides a competitive advantage, yes or no? Anyway, I don't know if you can swim very well, but even if the sea is near Athens, but I don't like a lot to swim. But let's speak about this fact now. Such is life. There is a story about 4 people named everybody, somebody, anybody, and nobody. Well, let's start. There was a very important job which had to be done, and everybody was told to do it. Everybody hoped that somebody would do it. Anybody could have done it, but nobody did it. Okay, somebody got angry because it was everybody's job. Okay, everybody hoped that anybody would do it, but nobody realized that everybody would do it. And it all ended with everybody blaming somebody when nobody did what anybody could have done. It's a trouble. It's not a trouble. Believe me. Why? Because in a center with a functioning quality management system, dear friends, everybody, somebody, anybody, nobody have specific names and know their area of work very, very, very precisely. And the quality management system, be sure, is not a staff punishment. Nobody can punish you. Why? Are you punished sometimes? Maybe. I was punished, and I'm punished, but it doesn't matter. It is an instrument for continually improving performance. But at least I can close with Aristotle that the quality is not an act, it is a habit. Thank you very much.